phosphate, not glucose 6-phosphate, into our catalytic enzymatic degradational process to consume glucose to produce glyceraldehyde 3-phosphate in the prep stage, and then push it into the payoff stage to get that NADH and those ATP moieties. So when we have a look at our main energy source in the sense of glycogen, if you're a human, in the sense of storage, gives us an accessible amount of carbohydrates when we need it to maintain, hopefully, homeostasis of the glucose levels, or starches in plants, and sometimes we utilize those starches also. We can actually utilize a enzyme, an alpha amylase, to take those particular starches. In this case, we'll use a starch to make saccharides. And what we're basically doing is taking our long chain. Remember, we have a 1,4 configuration. We have a non-reducing end. And in this case, it is a homo polymer of glucose. And we can basically start cleaving those particular simple bonds, those 1,4 bonds. And we do that by utilizing a glycogen phosphorylase. So it needs a source of phosphate, and this could be inorganic phosphate, or it could be materials coming from ATP, as we've seen in the previous reactions. And in this case, we will take off one of the individual glucose moieties from the non-reducing end, and then basically phosphorylate it, produce one glucose 1-phosphate. We then see a truncation with a new reducing end being established in the glycogen molecule, the starch molecule. So again, we're just cutting on off one at a time. And that's why we looked at amylopectin in regards to having multiple non-reducing ends and having that capacity to have multiple enzymes or one enzyme doing multiple work on different sites very rapidly, very fast to maintain that free carbohydrate level. So reducing that glucose 1-phosphate, which then can go through the process to give us the issue of producing glucose 6-phosphate, that goes down into then the glycolytic pathway. Some of the other monosaccharides we want to have a look at is fructose and galactose. Now, fructose is quite interesting because there's actually different kinases, hexokinases, in the body, and they have sequence homology, functional homology, but they have some different regulational components, and some of these are found in the muscle and the kidney. And it's going to come a little apparent later on why we have these differences, because we want to have different regulation and control. So what we see with these particular guys, let me just move this. We have basically fructose in regards to 3 different isotypes of the enzyme, muscle and kidney, and in liver. And in this case, we have a hexokinase using fructose and ATP to make the fructose 6-phosphate, or we have the fructokinase in the liver, which is going to be the main responsible thing to basically move material through. The liver is a very large sorting house of any dietary and drug metabolites or drugs that are coming into the system. Always goes through the liver first off, so it's called a hepatic clearance. So in this case, with the liver, we have fructose and ATP producing this time not fructose 6-phosphate, but producing fructose 1-phosphate. Now, both of these particular constituents, we can get to the issue of the fructose 6-phosphate directly moving into the glycolytic pathway, where the fructose 1-phosphate has to go through an additional step. And this is a reversible step. And it's very interesting because what it allows us to do is to take that fructose 1-phosphate and jump a couple of steps in the glycolytic pathway to go directly to producing the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. And as we know, the dihydroxyacetone phosphate by the triose phosphate isomerase is going to also produce glyceraldehyde 3-phosphate. So again, we get 2 moieties of glyceraldehyde and glyceraldehyde 3-phosphate. Well, we get glyceraldehyde 3-phosphate and glyceraldehyde, which we can then all go to triose kinase to also produce glyceraldehyde 3-phosphate. So we get 2 moieties ultimately of glyceraldehyde 3-phosphate from the fructose 1-bisphosphate by integrating 2 other systems of the isomerases and the kinase. So again, we have this capacity to keep drawing our material through to our main constituent, which is glyceraldehyde 3-phosphate. But I want to make sure you understand we can take glyceraldehyde, because as we know, glyceraldehyde and glycerol are also related, and therefore we can think about the metabolism. We think about triacyl fatty acids in regards to even metabolizing the glycerol backbone to produce glyceraldehyde 3-phosphate. So again, just something to keep in your back of your mind. So we have these particular systems to allow us to basically keep going and bring all our carbohydrates to that glyceraldehyde 3-phosphate constituent, which we can then feed into the payoff stage in the glycolytic pathway. Now, galactose is a very interesting one because there is a galactose disease called galactosemia, and it's actually not uncommon. And it's one of the diseases we actually screen in kids quite early with a heel prick to basically indicate whether they have a high level of galactose in the blood or in the urine. What happens is a galactokinase enzyme, and some people are deficient of this particular enzyme, and we can tell this by HPLC. So here we have a normal person with a heel prick or blood sample, urine sample, and we would see only a very small amount of galactose. But when we have someone with galactosemia, the galactose is off the Richter scale. And the problem is it can cause some metabolic issues in regards to mental retardation and also potential damaging aspects in osmolarity. So we've got to be very careful. But also galactosemia can also cause the issue of producing increased ocular eye pressure and cause vision problems, and also can lead to forms of blindness. So we've got to be very careful of that particular process. And what we do is just then limit the galactose by the dietary components. So galactose itself uses the galactokinase and the ATP mechanism to degrade ATP and give us that phosphate to make the galactose 1-phosphate. This particular one then goes into another system. We're going to talk about this system a bit more in the uridine diphosphate carrying system. And in this case, we're going to be utilizing NAD plus and NADH to go through an oxidation reduction reaction. Because ultimately what we're going to do, we're going to take this galactose 1-phosphate and use this UDP system like a figure 8, where we're going to be taking more galactose in and UDP glucose to start the priming system. And ultimately what we're going to do, we're going to be spitting out glucose 1-phosphate at the expense of consuming galactose 1-phosphate. So it's a way we can actually move things around and using those electrons and those electron transfer capacities to break and change bonds to give us the capacity to rearrange our carbohydrate to produce a moiety, in this case, glucose. So let's have a look. So here we have galactose 1-phosphate being present, and this primed UDP glucose, this uridine diphosphate glucose, basically giving this transfer group, this UDP transfer group, and allowing us to produce then glucose 1-phosphate. That glucose 1-phosphate can go through the various systems to produce then glucose 6-phosphate, which then can go into the glycolytic pathway. But what we are ending up with is UDP galactose. Now, this UDP galactose can also be cycled back, and we'll have a look at that in a second. But I want to emphasize importantly that we're going to see problems with this particular system in regards to if we don't have this UDP glucose transferase enzyme, so it's a galactose 1-phosphate uridyl transferase enzyme. Because if we don't have that particular presence, we're going to see that incorporation and increase of galactose 1-phosphate and not being able to produce the glucose 1-phosphate. So we're not so caring about the glucose 1-phosphate. It's this issue of the concentration increase of galactose phosphate and the backup of the metabolic pathway. We see a decrease in growth, liver damage, mental retardation, and usually death by the inhibition of this particular enzyme. We also have another component where basically we can take the UDP galactose and we can do some other things with it. And I'll show you in a second. But one of the issues is if we have that UDP galactose, we need another enzyme to produce this particular guy. And this is basically gives us the control of allowing us to move this molecule through the system and delimit the galactose 1-phosphate concentration. But this takes a cost in regards to taking NAD and making NADH and a proton by rearranging this number 4 position from the hydroxyl hydrogen to a double bonded oxygen in that number 4 position. We can then take this and metabolize this and do things with it. But if we don't have that UDP glucose 4 imparase, the number 4 position, again, we're going to have some problems. So again, I'm trying to highlight when we start seeing metabolic dysfunctioning in regards to the enzymes that we're working with, we're going to see a backup of a compound. That backup of that compound might leak into other pathways and start producing compounds that we don't normally see in a normal metabolic profile. And again, this is a part of the profiling system that we utilize in in disease discovery. So let's have a look at the UDP glucose galactose system in particular, because when we have that dietary control system, as I said, in many metabolic pathways, we have the capacity to basically channel material off to maybe a pathway that is not normally expressed or very low abundance. And that pathway might become then a major alternative route to getting away with the issue of the galactose 1-phosphate. So we've produced so we've gone back here. So we've produced this particular compound in regards to the UDP glucose and gone through the 4 imparase, and we can then metabolize that one to produce UDP glucose. Now, with that UDP glucose, we can basically get this to come off and produce glucose 1-phosphate and go to glucose 6-phosphate. So we have this capacity where we're consuming 2 moieties now of NADH. So we're producing 2 moieties of NADH by consuming 2 moieties of NAD+. So this enzyme works twice on the same molecule to give us a particular constituent of UDP glucose. Again, reflective of an alternative path that is not normally apparent unless you have a galactosemic situation where you cannot metabolize the galactose 1-phosphate, and this pathway then becomes a dominant feature in the control of your particular pathological disease. So we come to a situation where also in our normal bodies, we can have this issue of producing lactic acid from our degradation of carbohydrates. And again, these carbohydrates could be coming from glucose, from mannose, from fructose, as we've seen previously. And we get a payback situation where if we are not having enough oxygen being present in the muscles, particularly in like long sprints, or if you're a scuba diver, sometimes your breathing capacity might limit your oxygen availability as well as doing the exercises and everything else, of flipping your fins, you might get a cramp. That cramp is actually due to the buildup of lactic acid in the system. Now, as you can see here, we're producing our pyruvate from our normal metabolic processes, but without molecular oxygen being present, that pyruvate is not going through the normal route. It's going to be held and moved over to produce lactic acid, and that goes through the lactate dehydrogenase system. That consumes, importantly, an electron currency carrier in the form of NADH to produce NAD+. And as you can see, there is a preference for it to actually produce lactic acid, but there is a level of reversibility. So there's always a payback component. And this is a reaction that goes at negative 25 kilojoules per mole. So it doesn't increase in concentration until the rate of lactic production exceeds the normal factors that basically govern this particular process. So it's always kind of in there in the background, but because there's molecular oxygen being present, we're always going to get regeneration of pyruvate. But once that molecular oxygen decreases, we're going to start seeing the increase of that lactic acid. Now, lactic acid concentration is usually about 1 to 2 millimoles in a normal environment, but can exceed 10 times that amount into 20 millimoles with intense exercise. Now, any further increase in that and the relative abundance and maintenance of that particular type of concentration starts causing problems with pH. We already know what happens with pH in regards to protein conformational changes. But with the Z and D discs on the muscle fibers, when we talk about the muscle fibers moving, we have, and the actin and myosin interactions, we start seeing some changes that occur with there because of the pH, because of the lactic acid production. We're going to get some potential issues of tearing those particular constraints with those proteins being torn between each other. And that's where you get particularly, tears in your muscle and cramps, because that's what's actually occurring in regards to the situation. So we see the lactate dehydrogenase system as basically an electron sink in regards to giving us electrons placed in the lactic acid. So we're taking NADH, the electrons and the protons, and putting it in this particular, so we're going from this double bond to an alcohol, and in this case, putting electrons and protons in there. So it's kind of storing those in regards to being in a anaerobic environment. But we can't hold this for too long because of obviously the increased lactic acid causing damage. So let's have a little bit of a summary in regards to what we've seen in regards to this actually. This should have been in the previous slide. In regards to the galactose, so let's go back. Sorry, my mistake. That's lactic acid one should have been the next stage. But so let's have a look at the summary of the galactose metabolism to produce the UDP glucose. Now, as I said, the UDP glucose is a product of the normal pathway, and we have this priming reaction utilizing the product to then allow us to get to the final product of the glucose 1-phosphate. So that's the point I wanted to make. So it's like a figure 8 where galactose is coming through the system and needs this UDP glucose to be displaced to produce the glucose 1-phosphate, putting the UDP on the galactose, so it'll be galactose UDP, and going through the various imperases to additions of producing NADH. And that's where we're going to get that NADH to hopefully fuel the anaerobic metabolism also. So again, we're going to have some NADH circulating in the cytosol of the cell to be able to utilize that as a electron sink into lactic acid. So that's the connection between the two, with the galactose and the lactic acid. But again, when we go through the issue of anaerobic metabolism also in lower organisms such as in yeast and bacteria, we see a different situation occurring. So you see how pyruvate is produced normally through the individual yeast or bacteria, and it is decarboxylated. And this is a really important reaction, and we're going to study this a little bit later on today in regards to the removal of that carbon dioxide. So again, if this was a radio-labeled pyruvate and labeled in this position, and we had a fermentation under an anaerobic environment, we would actually see the radio-labeled carbon dioxide coming off. This particular system in the pyruvate decarboxylase will produce the acetic aldehyde or acetaldehyde constituent, and this is going to be then undertaken by the consumption of NADH, and again, using the protons and the electrons that's carried by this system, using the alcohol dehydrogenase enzyme to produce ethanol, but noting the issue that there's a level of reversibility. So when you consume ethanol, you are going to utilize your NAD plus, your alcohol dehydrogenase, produce a compound, acetaldehyde, and then go back to producing pyruvate. Now, for some people, there is a problem with the alcohol dehydrogenase. And so if you consume ethanol, alcohol, you can have some problems in regards to the degradation of this particular constituent. It can cause an increased susceptibility to alcohol poisoning and the effects of alcohol. This is common actually in some races, particularly in Asian individuals. I used to have a friend who'd give him one beer and he would be on his face because he couldn't metabolize, passed out, because he could metabolize the ethanol. And this goes through a particular enzyme system. And we're going to come back to this guy a little later on, the thiamine pyrophosphate, or the TPP. Because what we have this thiazolamine ring playing a particular role in sinking those electrons to give you this capacity to remove that particular carbon dioxide and rearranging the backbone to produce a carboxylic acid to an aldehyde to an alcohol. So remember, we have some particular systems that we are going to reoccur. So this particular ring provides a bit of an electron sink to give us the capacity to move things through quite nicely. So in the fermentation process, we're going to have this capacity occurring in the liver. So again, it's important why the liver is in a good function and capacity, and you can actually survive on a third of your liver tissue. So you've got 2/3 to waste. Hopefully, you're not going to be in that situation with cirrhosis by hepatitis or cirrhosis from alcohol abuse. But the main thing is the NADH, which is used in glycolysis, is basically regenerated during when we look at the mitochondrial respiration, which we'll see a little later on. And with that particular energy source, we're going to have, a normal metabolic process with the presence of oxygen. But in a hypoxic environment, meaning low oxygen levels, the NADH has to be basically regenerated to give us this particular cycle process. So again, this is the sink, the electron sink I'm talking about in regards to recycling that NADH between glucose, the payoff component in glucose. So again, we've got our preparative stage, now our payoff stage producing the NADH, and we're going to be consuming that in regards to this time not going through the whole process and depositing the same components from the pyruvate into the two forms of the lactic acid. So again, we have to be very careful in that. But we also utilize this particular process in fermentation. If you think of sharp cheddar cheese, what we are actually doing is basically inducing the fermentable products in cheese to produce lactic acid by giving it an anaerobic environment. So it actually has a change in regards to the fermentation process to our advantage to change the taste and the texture of foods given the presence of this lactic acid. You might have heard of lactose bacilli in your yogurts. It gives them a bit of a sour taste. And if you have your yogurt and you've kept it in the fridge a little too long beyond the expiration date, you might even find a more sour taste in regards to the increased metabolism of the available sugars and the available pyruvate and producing more lactic acid. So again, we've utilized these types of fermentational processes to give us an advantage in regards to flavor and taste of our foods. So we have also other decarboxylation systems which basically are going to target these types of bonds to give us this cleavage of that carbon dioxide. We see this in the pyruvate decarboxylase in ethanol production, but we also see this in two enzymes that we're going to study in the TCA cycle, the pyruvate dehydrogenase and the alpha-ketoglutarate dehydrogenase. And these are essential in the TCA cycle. So we're going to see this with the production of the acetyl-CoA, which will enter the TCA cycle. And we're going to see this occur in the TCA cycle itself at a critical state with that alpha-ketoglutarate, because we're going to cause the liberation of carbon dioxide. And it's this liberation of carbon dioxide which is coming from the backbone of those carbohydrates and the products in regards to the pyruvate, which is a 3-carbon moiety that's coming through, and it's going to produce a 3-carbon moiety in the form of acetyl-CoA, which is going to be decarboxylated by this pyruvate dehydrogenase and then continually be decarboxylated by the alpha-ketoglutarate dehydrogenase and some other systems and move it through into a cyclic system. But we're going to come to more appreciation of that in coming weeks after the second exam. So I'm just trying to see the whole overall process. So fermentation, we see the process of glucose, lactic acid, ethanol, arginine, pyrimidines, pyridines, ethylene glycol, which is not a good thing, that's commonly found in antifreeze. So this is what kills kids, because basically it produces a very highly toxic substance when it's metabolized by the liver. But what we see is a whole lot of different types of molecules coming through, and most of these we can handle if we're metabolizing phenylalanine, leucine, isoleucine, threonine. Sorry, leucine, not isoleucine, threonine and leucine. And then we have our base pairs with the pyridines and purines, whether it be DNA or RNA. And these also can cause some problems in regards to metabolism. We'll see a little later on in regards to gout, which is a uric acid, xanthine uric acid synthase depletion. So again, we have various products we can produce, which then can either be made into other things, or be, in this case, in carbon dioxide, be breathed out, or be utilized to balance pH. Remember, we talked about the erythrocytes and the carbonic anhydrase reaction with water and carbon dioxide, and the countercurrent system with fluoride to give us this capacity to change localized pH and give us bicarbonate or carbonate ions or carbonic acid ions to give us this capacity to buffer pH. So again, we've got to keep thinking about all the things we have learned in regards to the biochemistry. So we've come to our basic understanding in regards to the various enzymes that are going to be playing roles in this central key component, where basically we're focusing on this metabolismal process in regards to glucose, pyruvate, acetyl-CoA into the TCA cycle. There's actually a graduate course in ethanol production, and we do a little bit in lactic acid production, but mostly ethanol production. It's a graduate class that I run, which basically we work in a small microbrewery and a micro distillery. That class is run by myself and Nick, who you all know, and we basically go through and build a process to produce an ethanol containing product. It could be hand sanitizer, it could be alcohol, spirits, it could be beers, it could be wines, it could be meads, and we offer those to the community and look at the innovation, technology, and building of a startup. So that's a graduate class, and you're quite welcome to come to that class as a graduate student. So again, we're going to be looking more at this particular system elsewhere as well as this, but this is the main focus we're looking at in regards to glycolysis, which we now fully understand, to get into that pyruvate and see how this one in the aerobic condition, we're going to get that carbon dioxide, get it into releasing carbon dioxide and more carbon dioxide as we metabolize the carbon backbone of our carbohydrates. So that ends this particular lecture. We've looked at glycolysis. We're going to talk a little bit more about gluconeogenesis in the next lecture in regards to the process. And I want you to be able to ultimately compare glycolysis and gluconeogenesis, which we'll talk about in a second, and glycolysis and fermentation. Know and draw the pathways for preparation and payoff, indicating the products, including ATP and NADH. We're going to talk a little bit about the regulational pathways today, but also how we basically utilize glycolysis and the UDP systems that combat the concentration of galactose 1-phosphate and how we can metabolize galactose, and the difference between galactic acid formation in aerobic and anaerobic environments and fermentation, but also the process of where we are ultimately going to be getting our carbohydrates from our glycogen storage system. We're going to talk a little later on about glycogen storage and production of glycogen in regards to accessing that as an energy store. So let's have a look at our next lecture slides in regards to